Okay, guys. So let's begin. Turning on. Okay, we ready? All right, guys. So we stopped here. We were talking about the primary structure. The primary structure of amino acids are linked by a peptide bond. Remember, we discussed that. So we have an oxygen that has a hydrogen bond, and we have an NH3. So that looks like water to me. OH on one side and H on this side. So O from this side and two H's from the amino terminal. That comes out to water, and then that leaves. So you need to remember that condensation, we dehydrate, we remove water. You see the water moving away? That's NH3. Now it ends up as NH. So two hydrogens are lost. So one side donates two hydrogens, and the other side donates oxygen. That's important. The peptide bond, how is it formed? One side donates two hydrogens, one side donates oxygen, that's H2O. That's removed, then a peptide bond is made. So a polypeptide bond has directionality, sometimes called the polarity, and the amino terminal end is taken as the beginning of the polypeptide. So you see, that's the amino terminal, that's the beginning, and we're going down this route, and the end is the carboxylic terminal. So let's look at here. That's the amino terminal. You have that looks like tyrosine. Is tyrosine polar or non-polar? Just by looking at the structure of tyrosine. So let's solve it. Where is my alpha carbon? Is it this one or this one? The first one, because it has oxygen, the carboxylic terminal. That's the amino terminal. That's the hydrogen, and that's my R group. Does my R group have H or O to form a hydrogen bond? Yes. Do I only need an H and O to form a hydrogen bond? What if I had an N here? A nitrogen. That's also electronegative. So then the hydrogen combined with that. So if there was an NH3 here, so if there is another amino acid with NH, like this one doesn't have an O, it has an N. That N can also be electronegative. It can form a bond with the hydrogen. Clear? Okay. Now we said a disulfide bridge connect. So that's a one linear chain. That chain looks like a primary structure, yeah, or a secondary, that's a primary structure. So now we talked about one way where these primary structures connect. We talked about them connecting through a hydrogen bond, but now they can connect also through a disulfide bridge. Disulfide bridge. So most proteins can consist of 50 to 2,000 amino acids, and in some proteins, the polypeptides can be cross-linked by disulfide bonds. Please remember that, disulfide bond. So chain A and chain B is connected to each other by a disulfide bond. And this amino acid, so remember we said what is so unique about glycine and that it has two hydrogens. What is unique about valine? That it has, I'm sorry, not valine, alanine. It has an extra, it has a methane. Proline has a cyclic group. Now, there is one thing special about cysteine, has sulfur. So which amino acid has a sulfur in its R group? That is cysteine. And that's what helped us to make the disulfide, thank you, the disulfide bridges. So look at who's making the disulfide. It's the cysteine. You see the two S's between whom? It's between cysteine and another cysteine. Because who else has a cysteine? Only cysteine. So look at the first chain. You can have a horizontal bridge and you can have a vertical bridge. But it has to be between the two cysteines. So you see cysteine S connected to cysteine S. And then vertically between two chains, cysteine S and cysteine S. Clear? Now we move into secondary structure. So the secondary structure of the amino acids can fold into regular structures such as the alpha helix and the beta sheets. That is so important. I mean, we have to understand that the beta sheets, the beta pleated sheets, and the alpha helix are what kind of protein structure? Secondary. So the secondary structure is the protein structure that's formed by hydrogen bonds. So who achieves for us the secondary structure is the hydrogen bond between the peptide bond NH and the CO. When a peptide bond is made, does the water go in or water go out? Go out. Is that dehydration or hydrolysis? Dehydration. And when the water goes out, who donates the O? Who donates the two H's? Not the oxygen. It's this. So there are two amino acids, right? So one amino acid donates a whole oxygen. Now it has no oxygen, you see? And the other amino acids donates two hydrogens. That's now H2O. So now the one that has no oxygen, this one has no hydrogens, and then they have the red peptide bond. Really important. Okay. Now the alpha helix and the beta sheets and terns are prominent examples of secondary structure. They are a prominent example of secondary structure. So the secondary structure of a protein are the local interactions between the backbone of the two polypeptide chains. What bonding facilitates this alpha helix and beta sheets formation? Yes. Excellent. Excellent. You see, that's all we want. It's not that hard. So just the stuff that you need to know, and it's very easy. What is an example of secondary structure? Alpha helix, beta sheets. What kind of bonding? What kind of bonding is responsible for this alpha helix and beta sheets? Hydrogen bonding. And when a peptide bond is made between the two amino acids, one amino acid donates what, and the other has to donate what for the red peptide bond to happen? Excellent. Oxygen is one, and two hydrogens on the other, right here. Oxygen in one, this one gives oxygen, the other one, because remember, we need to get water out. So you can't say oxygen here, oxygen there, that's two oxygen. You can say oxygen here and two hydrogens there. H, two H's, one O, that's water. Yes. That's for a peptide bond. You see the red bond, the peptide bond? That's the peptide bond. Okay. The alpha helix is a tightly coiled rod-like structure, like a loop, with the outer groups bristling out from the axis of the helix. So it's spinning around like a coil. And all of the backbone, the carbonyl and the nitrogen groups form the hydrogen bonds, except those at the end of the helix. So the bonds are between the oxygen and by the oxygen and the hydrogen of the nitrogen. Between the oxygen and the hydrogen, because they're just like water. So you have O with an H. So that's how they make the bond. It's an electronegative bond. Oxygen, remember when they share, but the circle is going to be, the bubble is going to be so big around oxygen because electrons gravitate and spend more time with oxygen than hydrogen. You see, that's the loop. That's the alpha helix. So if I post a picture of that, what is this? This is a alpha helix. And they call it right-handed because you take your right hand, you stick your thumb inside the coil, and you see which direction are they which direction are they tilting. If that means they are tilting in the right direction. So that's what they are doing, right? They are tilting in the right direction. So like this, like a right hand coil. So that's the loop. You put your finger inside, you spin this way. So that means it's looping in that way, not the other way around. You see, that's the oxygen and that's the hydrogen of the nitrogen. That's how they make the bond. So the bond is between the oxygen. So when we make an alpha helix, not a peptide bond is already made. Peptide bond is to go from an amino acid to a protein to make a polypeptide. You see, that's the polypeptide bond. So you should be able to recognize if I give you a link like that and tell you, where is the peptide bond? You should be able to tell. You see right here, that's the peptide bond. It's between the carbon that donated an oxygen and became a carbonyl, and the ammonia that donated 2 hydrogens, and the link is right here. Now it's in red. If I change the color, you can still tell. So let's look at here. Yeah. Carbonyl, because now it has oxygen. When you have an oxygen double bonded with a carbon, that's a carbonyl now. It's a functional group known as carbonyl. Yes. Yeah, a peptide bond can only be between C and N, because who else can give me water? Who else can give me O and 2? Because remember, the peptide bond was made after I removed water. So if you want to make a peptide bond between this carbon and this carbon, there's no O here. And you cannot make it with the R group, because remember, the polarity has to be from the N terminal to the O terminal. So now I change the color red. If the color turns black, you still should be able to recognize it right here. Can you tell me where is the peptide bond now? I can put 1, 2, 3, and 4. Yes. Yeah. So it's which one? This one? This one is the, no, do you see a carbonyl? You have to have a remember, that's the carbonyl. It has to be between a carbonyl, carbon, and a nitrogen, because it lost the oxygen. Why did it become a carbonyl? Because it lost the oxygen and the electron went up here. Now you have a double bond. So you have a carbonyl because the oxygen left. And on the other side, you had 2 H's. You had 2 H's, they're gone. So now you have H2O and a bond between the N and the carbonyl carbon. So look for your carbonyl carbon. Where is the carbonyl carbon? That's the carbonyl carbon. And that's the N. So the peptide bond is between the carbonyl carbon. Okay, so you see, which one donated the oxygen? Which one donated the oxygen? So all of these are amino acids, right? That's amino acid 1, amino acid 2, 3, 4, 5, and 6. So the peptide bond is between the carbonyl carbon and the nitrogen, the carbonyl carbon and the nitrogen, the carbonyl carbon and the nitrogen, carbonyl carbon, nitrogen. Now, here, for this peptide to be made, it's already, we know somebody gave oxygen, somebody donated 2 hydrogen. Who donated the oxygen? This carbon or this nitrogen? The carbon. Who donated the hydrogen? How many hydrogens? 2. That's the peptide bond. Very good. Okay, now let's look at the beta pleated sheet. The beta pleated sheet does not look like a helix. The beta pleated sheet looks like a plate. It's another form of a secondary structure. So the beta sheets are formed by adjacent beta strands, And in contrast to the alpha helix, the polypeptide in a beta strand is fully extended. It is fully extended, and the hydrogen bonds link the strands in the beta sheet. So the strands of the beta sheet may be parallel, anti-parallel. So what is the binding between? It's between the hydrogen and between the oxygen and the hydrogen. Oxygen and the hydrogens that are adjacent. Just like the disulfide bridge, but now the disulfide bridge is always between two cysteines. Remember that? Between two cysteines. But in the beta plated sheet, it's just like that. But it's not between the cysteines. It's between here. You see this oxygen and the hydrogen of another chain, and the hydrogen and the oxygen of the other chain. So they connect vertically. They connect vertically, like here. That's a hydrogen. That's the oxygen connected with a hydrogen. Always the oxygen is red because it has all the electronegativity. You see? And now they could be parallel, meaning both of them are in the same direction. You see, those are going in the same direction. You have the same terminal pointing in the same direction, or they can be anti-parallel. Like the DNA strands are anti-parallel. They're not in the same direction. You have 5' to 3' and 3' to 5'. They are anti-parallel. There are also van der Waals, but the main ones are hydrogen. But van der Waals are there to support, additional support, to give additional integrity for the bond. But it's mainly the hydrogen bond. Remember, for secondary structure, right here, it's in black. Secondary structure is always hydrogen bond. Yes? You had a question? Okay. Tertiary structure. That is water-soluble proteins fold into compact structure with non-polar cores. That is now when you have amino acids that are polar and amino acids that are non-polar. When you throw them in water, the polar ones are going to be dissolving in water, while the non-polar ones are going to be hiding in the core, in the middle, away from water, because they are hydrophobic. That's how you get a tertiary structure. So the hydrophobic interactions. So low amino acids are grouped together in a complete protein, and the overall course of the polypeptide chain of a protein is referred to as its tertiary structure. So the tertiary structure of a protein is the three-dimensional structure of a polypeptide chain. That's what is very important to know. So secondary structure is secondary. The tertiary structure is three-dimensional. Secondary structure is two-dimensional. Primary structure is one-dimensional. Quaternary structure is four-dimensional. For example, an example of the tertiary structure is the myoglobin. So the interior consists almost entirely of non-polar residues, such as leucine, valine, methionine, phenylalanine, and the outside of myoglobin consists of both polar and non-polar residues. So, for example, for the exam, it'll help you a lot if you one day you can rely on your skill to identify the R group. Does it have oxygen? Does it have nitrogen or not to dissolve? But if you can go and you say, what are, because all of them, they are 26 amino acids. You could say, what are the polar amino acids? It'll give you the list. So who knows? Maybe when you look at the list, you're just going to know it. So me, when I look aspartic acid, I know that that's polar already. I don't need to look where is the R group, where that's the amino, that's the N, that's the amino, that's the O, that's the A. I don't have to do that. I just look at them. I familiarize myself with them. So some students tell me that. They said, I just looked at them. I kept looking at them, the non-polar ones, then the polar ones, and it stuck to my head. So now on the exam, you don't have to burn cells. So now there's no exam. You just look at them like a magazine, just every day. Amino acids that are polar, non-polar. You don't have to memorize them. They just look at them. On the exam, you will see them. You will be shocked. Oh, I know that one. That's polar. Oh, glycine. Oh, that's the non-polar. So you will know. Okay, so many proteins are composed of multiple polypeptide chains called the subunits. And such proteins are said to display quaternary structure. So the quaternary structure can be as simple as two identical polypeptide chains, and it can be as complex as a dozen of different polypeptide chains. You see, there are different globules. Talked about hemoglobin is the perfect example of a protein that an enzyme that functions at the level of the quaternary structure. But you know, earlier we said most of the enzymes function at the level of a quaternary. But aren't there exceptions? There's one exception. There's some exceptions. One that was working fine at third degree. Who was that? I just mentioned it. It has all of the amino acids in the hydrophobic core and the polar ones are on the outside. It was on the previous slide. Myoglobin. That's tertiary. You see, an example of a tertiary protein is the myoglobin.
You said an example of which one?
Of a tertiary structure that a protein that works as a tertiary structure. Okay. All right, we're done with that. Now let's look to the second one. Next one. Chapter 3. So now we're exploring proteins and proteomes. So milk is an example, is a source of nourishment for all mammals. It is composed in part of a variety of proteins. The protein, so milk is an example of a suspension because it has sugars in there, it has water, and it has fat. It has fat droplets in the milk. So the protein components of milk are revealed by the technique of the mass spectrometry, and which separates the molecules on the basis of their mass to charge ratio. So now we can separate it based on mass, but we need to know that the milk, the sugar of milk is known as lactose, and it is made up of glucose and galactose. Very good. So that's one component of milk. Does that mean milk has no proteins? Has proteins. Does that mean milk has no fat? That's fat, yeah. Okay, so the proteome, what's the proteome? The proteome is the functional representation of the genome. So the proteome is the entire set of proteins expressed and modified by a cell under a particular set of biochemical conditions. So unlike the genome, the proteome is not an n-varying characteristic of the cell. Proteome varies with the cell type, developmental age, and the environmental conditions. It's a varying characteristic of the cell. Proteins can be purified on the basis of differences of their chemical properties. And that's how we purify and separate proteins. So the proteome is different from the genome. And it is just a record of the set of the proteins that the cell expresses. We began the course by saying that we have a cardiac myocyte that's going to look different than a neuron, and it's going to look different from a hepatocyte, even though they all have the same DNA. That's a question that I can ask you. Why are all of these cells different? We said that the knife is sharp, it cuts, and the hammer is dull, it hammers. Both are made from steel, but the cells, you have a cardiac myocyte that's different from a hepatocyte, that's different from a neuron, but they have the same DNA. So why are they different? Yeah, because of the proteins. Why is that? I did that the first day of class.
Because only one section is activated, and the other ones are
excellent. Excellent. So the section that's activated is known as euchromatin, and the region that is not activated, heterochromatin. Very good. Somebody never went back and looked at their last week notes. It's been a while since last week. Okay. Okay. Proteins must be released from the cell to be purified. So cells are disrupted to form a homogeneous, which is a mixture of all components of the cell, but no intact cells. So we homogenize, we disturb the cell to form a homogeneous, which is a mixture of all the components of the cells, but no intact cells. So this is the homogeneous. We homogenize the cell, then we centrifuge it. When we centrifuge, we separate based on density. So the most dense will be on the bottom, and the least dense will be at the top. At the top is known as the supernatant or the pellet. That is the supernatant at the top. And in the bottom, you will have the pellet, So these are the fractions. The ones that, so when you homogenize and when you centrifuge the blood, the region that's all the way in the bottom, that's the red blood cell, that's your pellet. And the one at the top, your plasma, that is your supernatant. And the one that's between the two fractions, the buffy, creamy yellow coat, that's your thrombocytes and your leukocytes, that's also part of the supernatant. So the bottommost, that's your pellet. Okay. So proteins can be purified based on their solubility. So we know some of them have an R group that's polar. So that means it's gonna, the ones that dissolve, we can separate the ones that dissolve from the ones that don't dissolve. We can separate proteins based on size, like chromatography. So the big proteins will stay at the top because they cannot go through, while the little proteins are going to separate, while the little proteins are going to move through because they're so small. Then there is binding affinity. I can put, you know, like magnet, I can put, if I want a particular set of amino acids, I can put the opposite, like a strand, and make it pass by, and then they all gonna stick, and then I'm gonna be able to pull them out. So I use the charge, so I can have a positively charged amino acid to attract a negatively charged and pull it out from the protein medium that I have. That's how I separate. So we separate based on solubility, we separate based on size, we separate based on charge. There's also the concept of salting out. So salting out takes advantage of the fact that the solubility of the proteins varies with salt concentration. So most proteins require some salt to dissolve in water, a process called salting in. So we said earlier that in order to dissolve, you have to have electrolytes. So, and the water is electronegative, but you need, sometimes when you add salt, there's more electrolytes now. So it's not just oxygen and hydrogen. Now there's sodium and chloride, positive and negative. And that is what's going to help them solubilize more. So as the salt concentration is increased, different proteins will precipitate at different salt concentration, a process known as salting out. So the more we add, the proteins will start to aggregate and make like crystals. And when so when proteins dissolve in water that has salt, that's salting in. But when I add a lot of salt, now there's a reflux, the opposite. They start precipitating and make crystals, and that's salting out. Salting in and salting out. There's a video here.
In this video, you will learn about the concepts behind salting in and salting out. In biochemistry, these concepts are pretty confusing and people don't have a clear idea about this. So stay tuned at the end of this video, because at the end of this video, it will be totally clear to you. Let's say you have a protein solution. And with this protein solution, you're going to do two experiments. In the first set of experiments, you have added a salt solution where the ionic strength is not too high. And you have noticed that with mild increase in the salt concentration, the increase of the solubility. And there is no visible precipitate in this tube, and everything is soluble. In the second case, you have added salt solution, which has very high ionic strength. It's very concentrated solution. And in this particular case, you have seen without increasing the solubility, the solubility is dramatically decreased and there is a visible pellet inside as well. So if you plot a graph with solubility versus the salt concentration, you initially see an increase in solubility with increase of salt concentration. Later, with the increase of salt concentration, there is a decrease in the solubility. The first phenomenon is popularly known as salting in. With increase of salt concentration, there is increase in solubility. And the second condition is known as salting out. With very high salt concentration, the proteins are precipitated out. Have you ever wondered how this thing actually works at a molecular level? So in order to understand that, we need to know about the protein structure a little bit. So you know in protein structure, there are many ions, right? And there are many amino acids which could be charged. For example, there are aspartate or glutamate, which could be negatively charged. There are also possibilities that there are lysine, arginine, these kind of basic residues. And based on the pH, these residues would be charged. So protein would have some charge on it, right? It's totally legitimate. Now, salting in is the phenomenon where the increase in ionic strength is actually increasing the solubility constant or increasing the solubility of the protein. So how does that happen? So this effect is only and only observed in lower ionic strengths and different for different proteins. And a Debye-Huckel theory can actually explain the phenomenon. Let's look at it. So at very low salt concentration, let's look at the molecular interactions inside the solution. So this is the protein. Its negative and the positive charges are kind of masked by our salt counter ions. Okay. And now these salt counter ions are effectively masking all the different sort of charges of the protein. And then the solvent ion, in this case, the water, is actually creating a hydration shell around the protein. And you have more solute and solvent interaction than the solute interaction. As a result, the protein is nice, happy in this environment, and soluble. You don't see any precipitate. And you say the protein is soluble, right? But imagine a situation when you have added too much of salt. Let's say in this case, the salt is NaCl, and that's why you have so many Na+ ions. Now, this Na+ ion would love to interact with water molecules, and the protein is somewhere ignored. So, very less amount of water molecule is interacting with the protein and try to create the hydration shell around it and try to solve it. Rather, all of these water molecules are interacting with the sodium+ ion or the salt ions. So here, what happens in short is as the sodium ions are taking away all of these water molecules or solute particles, that means our protein particles are interacting with one another. And the solute-solute interaction is much more than the solute-solvent interaction. As a result, the protein gets precipitated. So that is the concept behind salting in and salting out. So in this situation, we are going to call it as salting out. And you can see the visible precipitate in the protein, right?
Okay. We talked about that. Okay. Now, dialysis. So proteins can be purified according to solubility, size, charge, and binding. So we talked about solubility. That's salting in and salting out. Dialysis. So dialysis is the salt can be removed from a protein solution by dialysis. So the protein solution is placed in a cellophane bag with pores too small to allow proteins to diffuse, but big enough to allow the salt to equilibrate with the solution surrounding dialysis bag. So now we will have the salt can be removed from the protein solution by the dialysis. So now we remove the salt, and that makes a lot of sense because sodium is so small, chloride is so small, it can leave because inside here I have salt and I have my protein. When I put a dialysis bag, the water is gonna make the salt go out, and then I can have my proteins at equilibrium. Then I say the filtration, gel filtration, and that is a mechanism known as molecular exclusion chromatography. It allows the separation of proteins on the basis of the size. So a gas column is filled with porous beads. When a protein solution is passed over the beads, Now you see the large ones, this is so important. The large ones stay behind and the little ones get out. And that's how we use gel filtration chromatography. So for all of these different methods, you should be able to give me an example. Proteins can be purified according to solubility. What process do we use? Salting in, salting out. Proteins can be purified according to the size. Using what? Using the gel filtration chromatography. What about the charge? There is ion exchange chromatography. So that allows the separation of the proteins. Remember when I said you give a different charge that tags them? So now the minus is going to stick with the minus, but the one that is the same charge is going to repel. So the ones that share, like let's say I want to separate a negatively charged amino acid. So I'm going to have to make my filter what? Negatively charged also. So that when I add my mixture of negatively and positively charged amino acids, the positively charged are going to stick to my filter because its opposite charges attract. But the ones I want to get are going to be repelled out, and then I'm going to get them out. So the beads are going to be, the beads in the column are made with a charge. Beads have charge. So when a mixture of protein is passed through the column, proteins with the same charge as the beads will exit because they're being repelled. You need to know this one, how it happens that if I need minus, and you need, remember we said that amino acids are charged, negatively charged and positively charged. So if I want the negatively charged to come out, my beads have to be what charge? -2. So that when I add my mixture of negatively and positively charged, the plus and minus are going to stick to the beads. So the beads are going to catch what I don't want. And then because what I want is the same charge is going to be repelled out. So they're going to exit the column quickly.
This is the charge, not the size, right?
Yeah, that's the charge.
So
The size was I had actual large, I had size to allow only, just like a dialysis bag. But now I have beads, but I'm not using a dialysis bag. Don't, and just set it as an example, but I'm not using a dialysis bag. I'm using beads. And the small proteins, the beads, they have small gaps between them. Big bead next to big bead, but there's small gaps that allow the passage of my protein that I want. So I separate between the charge, and I separate between the size with gel filtration, charged with ion exchange chromatography. Then we can have separating the protein with affinity. So now some proteins have a high affinity for a specific chemical or a chemical group. So now the beads are not made with a charge and they are not made with size. Beads are made with the specific chemical that the protein that I want, the amino acid that I want, likes to stick to. So a protein mixture is passed through the column and only proteins with affinity for the attached group will be retained. So the bound protein is then released by passing the solution through the column. This solution is enriched in the chemical to which the protein is bound. So here we see the glucose binding protein attaches to glucose residues, G. See, you see how now they're attaching yellow. The yellow is protein with my glucose, attaches to the glucose beads with the residue. And then I end up with the glucose binding proteins are released on the addition of glucose. So when I add more glucose to the solution, it's going to displace them, and then I'm going to get what I want. Now, gel electrophoresis. So proteins will migrate in an electrical field. Now that's based on electricity with a velocity v, directly proportional to the electric field E. So the charge on the protein Z and inversely proportional to the frictional coefficient. So the sodium, the disyl sulfate, that's an example of that is the SDS page. The charge, the size, and all the different ways. Okay, so that's the gel electrophoresis where proteins will migrate in an electrical field with a velocity. You see now they're moving. The slower ones will stay behind, and the faster ones will move. Now we can also use immunology, like in ELISA. So in immunology, immunology provides important techniques with which to investigate the proteins. So an antibody is a protein synthesized in response to the presence of a foreign substance known as the antigen. So I make an antibody against a certain antigen. So the antibody recognizes a particular structural feature on the antigen called the antigenic determinant or the epitope. So the place where the antibody binds the antigen is known as the site known as the epitope. Okay, so the monoclonal antibodies with virtually any desired specificity can be readily repeated. So immoral cell lines producing monoclonal antibodies can be generated by fusing normal antibody producing cells with cells from the type of cancer called multiple myeloma. And then a monoclonal cell line is isolated by screening for the antibody of interest. Okay, so let's go over this next week. Okay, let's stop here now at slide number 13. We'll finish up next week, and then the week after we're going to be tested on the material where we stop next week. Let me take the attendance, please. Please hold on. I'm sorry. Excuse me? Yeah, no, that's different. I have to still do the attendance and submit it in a paper.